Um, I'm excited to introduce them today. I'll start with Alex, who will be giving the first talk. He's a specialist in quantitative microscopy techniques, and he's doing some really fascinating work. He got his PhD in chemistry at Durham, and then moved on to a postdoc where he initially had a JSPS postdoctoral fellowship in Japan. And he's currently at York, where he is doing postdoc research into the role of protein condensates as sensory or metabolic regulators in plants. And so today he's going to tell us a couple of things. One, about how to track polycomb regulators, and two, whether or not plants are like light switches. So I'm excited to find out the answers to those questions. Alex, please take it away.
Fantastic. Thank you so much for the invitation. So, yeah, I guess many of you will be in the spooky spirit today, but some of you might be looking forward already to scenes like this in the spring, like this cherry blossom. So I'm here to talk to you today about whether this flowering is like light switches, and how we can track polycomb regulators with something called SlimVar microscopy. And the cold that's setting in now is exactly the reason why we can expect those blooms in spring. And the reason for that, because flowering is a good example of a switch-like life cycle event. It has very specific timing, as you know, because the fitness cost is very high. If it's too early, the plant will freeze and die. If it's too late, it'll be outcompeted. And there's a lot of parallel examples throughout biology. So when it happens, when the flowering happens, depends on the external temperature profile. As we know, it's been recorded for centuries for this particular example, over a much longer time span than the flowering actually happens. So the external temperature profile over dozens of individual cell replications is telling the plant when to flower. So the question we need to answer then is where in the plant is that information stored until it can be used to make that switch? If we can answer that, then plants could tell us a lot about how other eukaryotic organisms, including ourselves, either remember or potentially switch that differentiated identity throughout their tissues through their gene regulation. So I want to acknowledge the people that helped me do this work. The wonderful folks include an interdisciplinary collaboration between my group under Mark Leake at York and the John Innes Centre in Norwich, UK. So today's talk, I'm principally going to talk about my development that allowed these discoveries with Mark Leake and our specific results for plant chromatin regulators with Caroline Dean's group. But it's also very much motivated by a really valuable modeling work by the neighboring group at John Innes Centre led by Martin Howard. So chromatin, as we currently understand it, is actually quite an incomplete model of memory storage. And the main problem with it is that in a differentiated cell state of a cell, it must be incredibly stable against cell division. And almost everything we know about epigenetic storage is to do with the histones or the DNA itself, which have to be split between daughter strands during replication. So after a few replication events, you'll naturally lose half of the memory or more each cycle. And then you're depleting that state, which before it can be repaired. So there's an indiscriminate reset of expression every time this happens. But there are other accessory factors, including nuclear proteins helping the nucleation and spreading of the histone markers that define the epigenetic state. And these don't necessarily get diluted during replication. And protein assemblies, as we know throughout biology, can exhibit very strong positive cooperativity, either as affinity or avidity. So based on the Howard Group's work, our hypothesis is that a particular locus, a self-sustaining protein assembly, could be kind of a bookmark for these missing histone marks. And it just so happens that in Arabidopsis, which is our model plant, there is a single gene bottleneck induced by temperature over a long period of time to transfer from a vegetative to a reproductive state. And it might seem a little bit odd, but we actually need to look at the root tips to answer why flowering happens. We need to zoom into the easiest part of the plant to image FLC, flowering locus C, at a molecular scale, and that's the root tips. And it just goes to show that this really is individual events at each meristematic cell, because these form really clear files, vertical files from the meristem at the bottom of the root tip. And you can see as we have a progression of cold, eventually some of these files get turned off until all of them are turned off. So it really is like a light switch, except it's not the flowering light switch for the whole organism, it's at each individual copy, genetic copy of FLC in each meristematic cell that is affected independently. So this also tells us FLC acts as a digital switch because there aren't really intermediate states. You can see that there's kind of a halfway state before it gets turned off, but that's because FLC has two copies. So there's originally two copies on, then one turns off, and then both turn off irreversibly over time. So modeling tells us that's a consistent kind of tug of war effect with these very strong feedbacks, either to an on state or to an off state, that depletes any intermediate switch. And this is a process of nucleating the silencing markers for FLC by Polycomb. So what are the candidates for these protein accessories that I was talking about? Well, VIN3 and VERN5 are known to be induced by cold, as opposed to a separate pathway for silencing FLC through the autonomous pathway. So if we look at the specifics of these two proteins, we know that there's a huge amount of biochemical evidence that shows that they localize to FLC at the appropriate time. They recruit Polycomb to the gene, and then they cause these silencing markers to spread across the gene as a function of cold exposure. And then that silencing state sets in and persists after return to warm. So this is a really good set of candidates to look at. It also tells us that the nucleation and spreading are separate processes. And if you interrupt this at any given time, the activation markers start to reappear and the flowering is significantly delayed. So if you model this knowing the experimental rates from these biochemical experiments, you can predict how many units of the accessory protein would hypothetically be needed to stabilize this memory. So it's a really important input from Martin Hauer's group here, where you can model this and you know the individual rates, and you can predict the number of proteins in a particular assembly were they to meet FLC at the particular appropriate time. The minimum threshold for that would be an assembly size of around 10 to 17 proteins, which is very much feasible. So if we really want to look at this in detail, we have to look at the mechanism of how these two proteins not only meet FLC, but also how they assemble. And they're known to have this kind of head-to-tail polymerization domains where they can interact with themselves and with each other, and then localize to FLC. So what are the kind of biophysical, this is my part in this story, what are the biophysical techniques that can actually tell us, that help us see these assemblies? Well, the obvious go-to is confocal microscopy, where there's clearly expression, but not much indication of assemblies because they move too quickly. However, as we go to higher resolutions and faster images with AeroScan, we can see some hints of these forming assemblies, but this clearly needs a step change in the approach that we take. So what we look at in my lab and Mark's lab in York is single molecule, single particle tracking with molecular precision. So there's specifically a form of single molecule localization microscopy that is for particles in vivo that can beat the diffusion of those particles. So you can image resolved particles in a single frame and then track them through subsequent frames. Not only can you do that, but if you take fluorescent proteins in an endogenous level, you can bleach them sequentially with a high power laser. You can actually measure the steps of single molecules in these individual particles over time. So you can not only track them, you can also count the number of proteins, which is of course perfect for this question. And so this all happens on a very, very fast time scale, around 1 to 20 milliseconds. And so this is also an endpoint experiment because of photo bleaching all of these. So what you see is, if I go back to the previous slide, you can compare this. You can then see that this photo bleaches them extremely rapidly. And you can actually resolve the individual particles that each contain maybe just a few or a dozen molecules of these proteins. And the reason this works so brilliantly well is because we've changed the way that the microscope is set up to mitigate those aberrations that you would normally see for single molecule tracking at depth in plants. This is about 20 microns into the plant, and it's an individual nucleus of an Arabidopsis plant cell, in which these can then be repeated at different exposures in separate replicates at separate exposures to cold and so on. So what happens when we quantify these, if we track each individual particle and count the number of molecules in each particle? Well, as a function of time, you can actually see that the Vern5 already is constitutively expressed, but it increases in expression over time, not only as the number of Vern5, but the number of Vern5 per assembly that we track. So it's clearly an increase in the stoichiometry, the number of particles per assembly. And that's a key difference to what was known before. Vin3, on the other hand, increases transiently and then drops away. So this threshold for state switching that was predicted by the model, this is actually in exactly the right range that was predicted by Martin Howard's group. If we look at this in a kind of orthogonal way, so we're looking at the Vern5 number of particles per assembly, number of molecules per assembly, you can actually see that we can see individual subunits down to dimers and individual molecules, but we can tell that these are particles made of individual head to tail polymerized dimers of Vin5 and potentially Vin3 as well. So this is very much in line with our expectations. So to go even further than this, what we did was we expressed a LACI YFP expression system and a LACO reporter for the FLC gene itself. So we induced LACI YFP, which then stuck onto FLC. And then we did a Z-stack to find FLC before we then did two-color tracking of Vern5 labeled M scarlet. And what we found was that these two actually co-localize at late stages of vernalization, therefore showing that not only do we have the requisite number of molecules, but these also appear at the FLC during the period of time in which it's conducive to silencing. And you can see that a quantitative analysis shows that these co-localize and track over time. So the way that we quantify this is to take these kind of videos. We can literally see the co-localization when we register those videos. And then we count not only how many Vern5 molecules are at FLC, but also how fast those two move. So we can see that the diffusion coefficient, and these are basically code for non-vernalized, 2 weeks cold, 6 weeks cold, and after cold. So when we go through cold, we then collapse the Vern5 diffusion to the same level that we saw for FLC. So those are co-localizing and the Vern5 is being immobilized onto the FLC in part. But as I mentioned, we can count the Vern5 molecules. And what's really interesting about this is that Vern5 does co-localize to FLC even before cold, but after cold and during and after cold, which indicates a persistence of memory over several weeks, so several cell replication cycles, you can actually see the localization of Vern5 at a higher number of molecules per assembly. So larger assemblies of VERN5 above the predicted threshold are localizing at the gene during the time when I expect it to be silenced and maintained in a silencing state. And we can quantify the percentage of those when we take different thresholds. And it seems that a threshold of around 6 to 10 molecules is sufficient to explain this behavior. So to summarize, basically, we've found a correlation between larger assemblies, which are known to be chromatin regulating proteins, acting collectively at individual FLC sites, individual genomic sites, which is then correlated with the silencing as a whole. So we haven't probed the individual silencing behavior, but we have shown a strong correlation. The only other thing is that we haven't yet done this as a function of replication. So we haven't shown it before, during, and after replication, because I mentioned this is a specific endpoint experiment. But what we have done is shown that it's conducive to this new model for how chromatin regulators support the regenerative nucleation and spreading of chromatin silencing markers. So to summarize, the key kind of take-home messages are Arabidopsis in plants in particular are a great system to study epigenetic marks. And it suggests that memory is not only encoded in the methylation of chromatin itself, which would decay otherwise over a few replication events, but also in the protein cofactors that regulate it. And the key thing is that this additional memory elements only work if these cofactors form assemblies of a certain size due to strong positive feedbacks. So our biophysical imaging and tracking approach discovered those assemblies, quantified them in detail, correlating the number of molecules and how fast they move, and showed that they roughly meet that model size threshold. So thank you for your attention, and any questions, please.
Thanks so much, Alex. That was super cool. To the audience, if you have any questions, feel free to raise your hands and I can unmute you, or you can type them into the Q&A for Alex to answer now or as the other talks are going on. I'll start by asking you, you mentioned at the end there that you haven't tested it through replication, but can you hypothesize whether you think these protein assemblies are remaining associated with the FLC locus as the cells are replicating, or how do you think this silencing is maintained for such a long time?
Yes, so the key kind of open question about this is exactly that, is how long do those individual assemblies, now that we can see individual assemblies, what is the length of that interaction between those assemblies and FLC? Is it kind of a, a Lego model where they bolt together and then they stick on the FLC and they persist there? Or is it more of a kind of tumbling kind of traffic jam model, a bit like you might hear described for protein condensates, where they kind of come together transiently and then leave, but then the assembly as a whole persists throughout replication? And that's still an open question. It's a very good question indeed.
So then do you think like the assembly of these complexes, I'm curious about how active it is or how dynamic it is when you do the bleaching experiments. Can you observe recovery of the fluorescence, and does that also tell you anything about how dynamic, how much switching there is between the individual monomers of these assemblies?
I think if it was a system that was easier to image, we'd probably have enough bandwidth to see that. The reason this was quite difficult is because it's in the meristem of the living root. So we currently have only just enough photon budget to be able to see them localized rather than turning over. It's to do with this timescales that I was talking about. So typically each frame would be maybe 10 milliseconds, but then because we're using up that photon budget to track them very quickly, they might only last for another 10 to 15 frames, so we know that the interaction at least lasts as long as that, but obviously compared to the replication time scale, that's quite short.
Sure. Well, I took up a little too much time with my questions, so you've just gotten a question in the Q&A. If you wouldn't mind answering that as we transition into the next talk, you can go ahead and answer in the Q&A box. But again, thank you so much, Alex, and we
Thanks
will
very
now
much.